The overall goal of this cell isolation procedure and migration protocol is to show a reliable way of isolating endothelial progenitor cells and their migratory potential towards serum samples of cardiac surgical patients.
This method can help to answer key questions in the regeneration of the endothelial lining in blood vessels, which is needed after cardiac surgery due to ischemia and reperfusion-related injuries. The main advantage of this technique is the relatively simple way of isolating progenitor cells, including the pre-isolation of CD34 positive cells.
In addition to this, major complications after cardiac surgery remain too common, underlining the need to identify deleterious and protective mechanisms during cardiac surgery.
Endothelial progenitor cells are crucially involved in the neovascularization of ischemic tissues and known to provide cardioprotective properties.
To begin the experiment, mix blood 1:1 with calcium-free and magnesium-free PBS. Add 15 milliliters of density gradient solution to a 50 milliliter tube. And slowly layer the diluted blood on top of the density gradient solution. Next, centrifuge the samples. Using a sterile plastic pipette, carefully collect the buffy coat layer of every tube and place it into another tube while avoiding the density gradient solution.